Hello friends, in this video we are going to discuss about the second step of translation in prokaryotes, that is elongation. This we will discuss in two parts, part 1 and part 2. So first of all, what is elongation? It is the extension of polypeptide chain by adding amino acid one by one to the growing polypeptide chain. It is the second and crucial step in translation. This process is facilitated by several elongation factors, EF and ribosomes, which moves along the messenger RNA molecule codon by codon. So let us see these elongation factors, or in short EF. Elongation is aided by three protein factors. They are EFTU, EFTS and EFG. Now let us see them one by one. EFTU, that is elongation factor thermo-unstable. It delivers amino acyl transfer RNA, that is transfer RNAs carrying amino acid to the ribosome's A site, in order to ensure correct codon-anticodon pairing. It requires GTP for binding to transfer RNA and hydrolysis of GTP. Upon correct pairing, that is codon-anticodon pairing, release of EFTU GDP takes place. Now second is EFTS, elongation factor thermostable. It helps recycle EFTU by exchanging GDP for GTP on EFTU. That is, regenerates EFTU after the latter has yielded the energy contained in attached GTP molecule. Now third is EFG, that is translocase. It mediates or facilitates translocation of the ribosome along the messenger RNA, moving the transfer RNA from A site to P site and from P site to E site, when the translation initiation complex is formed. This we have already discussed in initiation video. You can refer my video, the link is given in the description box. The transfer RNA binding region of ribosome consists of three compartments. First is A site, that is amino acyl site. It binds incoming charged amino acyl transfer RNAs. Second is P site, that is peptidal site. It binds charged transfer RNAs carrying amino acid that have formed peptide bonds with the growing polypeptide chain, but have not yet dissociated from the corresponding transfer RNA. Third is the E site or exit site. It releases dissociated deacylated transfer RNAs so that they can be recharged with free amino acid. This is the ribosome and these are the A, P and E sites of ribosome. Now there is one exception regarding the translation. Initiation codon AUG is positioned in P site, hence all except initiating methionine transfer RNA, FMET transfer RNA FMET occupies P site at the beginning of the elongation phase of the translation directly. Hence vacant A site is ready to accept the transfer RNA for the next codon after AUG, and in this way elongation takes place. Now let us understand this elongation in brief with the help of this flowchart. Elongation begins with the binding of second amino acyl transfer RNA at the ribosomal amino acyl site or A site. This is escorted by elongation factor carrying GTP, that is EFTU. As the transfer RNA binds, GTP is hydrolyzed and EFTU is released. Then a peptide bond is formed between N-formyl methionine transfer RNA at the P site and second amino acyl transfer RNA at the A site, and this peptide bond formation is catalyzed by the peptidal transferase. Then N-formyl methionine is transferred to amino acyl transfer RNA at A site, forming a peptidal transfer RNA at that site, and an uncharged transfer RNA is left at the P site. After that, peptidal transfer RNA is translocated to P site and uncharged transfer RNA is translocated to E site. A site becomes empty, that is unoccupied. Then new amino acyl transfer RNA bind to A site and process continues in a cyclic manner till entire messenger RNA is read or decoded. Now let us understand this elongation step or phase with the help of this diagram. These are the A site, P site and E site of the ribosome. As we have discussed earlier that initiation codon AUG is positioned at the P site of the ribosome. Hence, the initiating methionine transfer RNA comes and binds to this P site, as a result of which A site is empty, which is then ready to bind with the transfer RNA carrying the second amino acid, and in this way elongation begins. So the amino acyl transfer RNA for the second codon comes and binds to this A site, and this is escorted by EFTU GTP, elongation factor thermo unstable. As soon as this amino acyl transfer RNA binds to the A site, GTP is hydrolyzed and EFTU is released, which has GTP. Then EFTU GTP is regenerated by the EFTU EFTS exchange cycle, which we will discuss in detail in the next video. So what we see that initiating transfer RNA carrying the N-formyl methionine is present at the P site and the transfer RNA carrying the second amino acid is present at the A site. Then the peptide bond is formed between these two amino acids. This formation of peptide bond is catalyzed by the peptidyl transferase enzyme. After that, N-formyl methionine is transferred at the A site. Hence, peptidyl transfer RNA is formed at the A site and the uncharged transfer RNA is left at the P site. After that, peptidyl transfer RNA is translocated from A site to P site and uncharged transfer RNA is translocated to the E site. This then exits from this E site. Now the A site becomes empty or unoccupied, which then binds with the new amino acyl transfer RNA depending upon the codon that is present in the A site. So the process continues in the cyclic manner till the entire messenger RNA is read or decoded. So this is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.